Yeah, so I think that this is well known that there are many proteins that are associated with diseases somehow, but we haven't been able to still use those opportunities to develop therapies, and it's because many of them are just difficult to handle with a drug-like molecule, and really the allosteric drug discovery field, which is just exploiting, is aiming to transform that, as it's been said before. And what we have is a tool to do that, and which we have validated in several programs, as you will see. What we have is a method is based on molecular simulations and physics that provides quantitative predictions of binding hotspots. And from that, we can discover molecules using supercomputers and the information about the structure of the protein. And this is something that we have been developing for a number of years. So the technology is fairly mature, is validated, and still improving because technology is growing so quickly. We always add new features. So if we go to the next slide. This is, I think, now redundant with what we have seen, thanks to the introductions by Dr. Cunha and Dr. Brother. But just to insist that this is what we faced in the very beginning, in the very early days of the company. We didn't want to hit the orthosteric binding site, even though our targets had those sites, because we knew that it could give us an advantage if we could avoid competing with substrate. We could then find new properties, new molecules that had completely different properties. And if you go to the next slide, this is just a picture of the different things that you can do by targeting the allosteric binding site instead of the orthosteric. You can stabilize the protein. This is just by binding to the protein in the allosteric binding site, you make it more stable, and you can have a gain of function. That's where the company's name comes from originally. That's been our primary objective for some years. But we have actually now expanded that. You can also destabilize in the same way by targeting a conformation that is not the native conformation of the protein. And a particular form of that is actually, and this is a really exciting development, I guess most of you have heard about targeted protein degradation with the PROTACs approach. The PROTACs just needs a molecule that binds to a protein, just it's like a handle to the protein. It doesn't have to touch anything that is functional. So any protein that doesn't have an active site, if you can find an allosteric binding site and a molecule that binds to it, you can destroy the protein, not just inhibit it as before. You can completely destroy it, and this has a very profound effect. And then we have the classical allosterism that Professor Cornea presented. When you look at systems in vitro, you can see that you can inhibit, and actually using the same site, you could activate as well. And this is wonderful because it provides a huge variety of more subtle ways also of modulating your target than just an orthosteric drug just goes there, inhibits, makes the protein inactive. But this is a broader range of functions that you can achieve. So that's what we are now looking at, the whole picture. So if you go to the next slide, please. How we do that? We need the structure of the protein, and thanks to the incredible advancements in structural biology, and more recently in computation and AI and the AlphaFold program, that's essentially the whole of the human proteome. So almost any protein we can work on. And then first we have to discover the binding site. Then we discover molecules that bind to it. Then we have to, of course, do the experiments and validate that it's the case. and then we have to develop those molecules into candidates. Let's see each one of those 3 steps a little bit more in detail. If we go to the next slide, please. It's actually a movie where, I don't know if, yes. So this is just to provide more of a visual explanation of what we're doing. Actually, the core of the technology is molecular dynamic simulations, where in the supercomputer, we simulate the protein that is moving, and it's actually interacting with water molecules and with organic solvents. And we can simulate this for a relatively long time, and then we can see. Sorry, I'm seeing another movie now. I don't know if you're still in the first one. Sorry about that. Yeah. So what we can see is if the protein prefers to interact with water or with organic molecules. And this, which now obviously is very visual in what you were showing, actually we have complex algorithms that analyze this and extract from that binding free energies that identify the most important binding points on the protein surface. So if you go to slide number 46. Yes, no, the previous one, 46. Okay, this one, yeah, and for this one. So this is a bit more detailed explanation of exactly what we do. We start with the structure of the protein. And as I said, the source, it can be anything. It can be homology modeling, it can be AlphaFold model, it can be a X-ray structure or cryo-EM model. Then we have to obviously analyze and check that everything is properly, there's nothing wrong there. Then we can run the simulations that you were seeing before and extract this essential information about binding hotspots. And importantly, then we have a third step where we apply our algorithms that are proprietary and subjected to a patent, where we actually quantify the exact binding free energy that you can get by placing a particular atom type on each point of the protein surface. And then finally, we have a clustering algorithm that identifies allosteric binding sites as a collection of binding hotspots. So this is the definition, doesn't rely on shape, it doesn't rely on anything else on these physics-based simulations, and tell us quantitatively if you have a binding site that is druggable. And this is quite unique and opens the door then to identify molecules that also bind to this. And this is the next step that now you should be playing the second video, where you will see exactly here the binding site that we have identified in this previous step. The dots there represent the key binding hotspots that we have also identified. And then we screen millions of compounds, and soon we will be working with actually billions of compounds that we match against the protein structure, and particularly we look at which ones of them can fulfill the binding hotspots we have identified before. So we end up selecting a very small subset of these molecules. You can stop the movie now and go to slide number 48. Thank you. And these are the ones that then are going to be selected. The process is a bit more convoluted. Again, this is the picture. The movie gives you an idea of how this works, but it's more convoluted. We look at different atom types that can be how they interact. We can look at the flexibility of the protein in more detail, and we are also using other methods after docking to improve the results, methods that we have developed, by the way. And finally, what is important is we end up selecting a very small set of molecules, just in the order of 100 or so, that then they will be experimentally tested. And so this takes us to the next slide, which is that, and just with the multiple projects that we have done in the company over the years, the average time that it takes us to decide if an allosteric, if a protein contains a druggable allosteric site is 1 or 2 weeks only. So that's very, very quick. And then the virtual screening of about 10 million molecules is less than 1 month. So actually, the majority of the time is just setting up the assay and testing those compounds that we select. And on average, for binding sites, and remember, these are always binding sites that never has been targeted before because people did not even know that they existed. We have had 14% of success. That meaning that if we buy 100 molecules, we have 14 that are active. So which is plenty to start hit analoguing and then the evolution of those compounds and start the drug discovery project. And that's at the very least more than 1,000-fold better than just for doing the random screening by high throughput screening, for instance. So then if we go to the next slide. Yes, and this is very important. So we have obviously emphasized the computational technology because this is something that is quite unique, but the company is very much real. It's not virtual. Once we have compounds that are active, we have to do the drug discovery process and look at the science in detail, understand the mechanism of action. With biophysical techniques, we have to do SAR with medicinal chemistry, understand how we can improve the properties of the molecules. We support that with computational methods as much as we can, but of course, there's medicinal chemistry, hardcore analysis of what's happening and improving those molecules, and structural biology, of course. And in the end, biology is king here, so we have to follow always a biological signal. We have to have a therapeutic effect. So it's not just a matter of binding. And this is what I want to emphasize in the next slide, is that allostery, of course, always computation has to go hand in hand with experiments, but in allosteric drug discovery, this is particularly true. If you look at the first panel to the left, this is one of our molecules in an enzymatic activity assay. And the flat line means it's doing nothing. So it's a silent binder. We know that it binds because we have other techniques that and other experiments I'm not showing, but we know it's binding, but it's not doing anything. If you look at it just for the protein in isolation. So it's binding elsewhere, it's allosteric in that sense, but it doesn't have an impact as on the enzymatic activity. But what happens in the cell, that's what you have in the second box in the middle, is actually, you can see a dose-dependent increase of enzymatic activity. And that's because this molecule is binding to the protein, is making it more stable. It's actually increasing the amount of protein that you have in this case is in the lysosome, and therefore you have more function, more effect, right? You actually double the amount of enzymatic activity. Of course, the time is different. So this is an experiment that is over a period of several days. It's in the right biological context and so on. And then the most important thing is you have to look at the downstream consequences of that. If you're having some consequence that can have a therapeutic effect. So in this case, and you'll see more of that later, we managed to reduce the amount of alpha-synuclein, which is a protein that is the root of the pathology of Parkinson. Right? So this is a message that is really, really important. In allosteric drug discovery, the effect is a lot more subtle. You cannot skip at all the biological part. It's a key component of what we do. And with that, I come to the conclusion. So next slide, please. And just to reiterate, CTX makes allosteric drug discovery fast, efficient, and rational. And this has given rise to actually a range of projects, both internal and with collaborators. And Manolo will be showing some of them in the next presentation. As a result, CTX has been thoroughly validated. And this is transformative because, as Dr. Cunha and Dr. Brother pointed out before, allosteric drug discovery has huge potential, but rational discovery of allosteric drugs is just not accessible. And we are pioneers in this field. And we continue to improve the technology, adding new features and capabilities to find allosteric drugs even faster and more efficiently. There are terrific advances in almost all aspects of science. And things like the incredible growth of chemical space, which is now reaching this 40 billion space, and that you can buy very quickly and for a low cost. And of course, artificial intelligence, not only on the structural prediction, AlphaFold, of course, is a primary example of artificial intelligence, but also to predict toxicity of targets, the MPK properties, etc. This is a huge help in drug discovery. And computation, of course, is essential to reach successful allosteric drug discovery. But it is also essential to recognize that allosteric drugs modulate biology in ways that are not obvious. And the discovery of allosteric drugs must go hand in hand with relevant biological experiments. The biology team at CTX, the chemistry team, the pharmacology team are just as important for gain as the computation or the structural biology. And we are very proud of what we have achieved together. Thank you very much.